Hey guys, this is my presentation on the yellow fluorescent protein. My chosen gene is the Vibrio fischeri yellow fluorescent protein. So this protein is a bioluminescent bacterium which is found in squids and other fishes. It can be seen as a biosensor technology in food, specifically in yeast. The Vibrio fischeri protein can change the color of the bioluminescence emission of the luciferase, which can only happen at temperatures below 20 degrees Celsius. And actually, this luciferase enzyme is what the fireflies and squid have in common. For fireflies, I think it was meant to show the energy transfer in the cell as well as the cell respiration. This also affects the antennas. It doesn't make their antennas light up, but I'm not sure what it affects. It just affects their antennas in some way. And to give a little background information about the bobtail squid, these amazing cephalopods have a symbiotic relationship with the marine bacterium Vibrio fischeri. This relationship is known as symbiotic bioluminescence. In this relationship, the adult squid becomes infected with this bacterium via their light organ. Their light organ would be described as something kind of similar to the squid's own eyes. Now basically, the relationship of this is the squid gives this bacterium a home and food, and in turn the bacterium, through its yellow glow, allows the squid to camouflage with the moonlight under the waves. Now to continue, here is the full gene sequence along with the highlighted enzyme codes. The highlights will be talked about in a minute, excuse me. As for the table below, my E value, which is responsible for the number of results expected to see when searching the DNA sequence, is 0. The identity describes how similar the DNA sequence is to any of the results. So like the exact matches. Since I only had one, it shows 100%. As for the GATs, which is not shown on this slide, I don't think, no, it's not shown on this slide. The gap percentage is responsible for the gaps between the DNA sequences. This number is, for my gene, is 0. Now on to the next slide. The initial plasmid I chose is YRP7 with the enzyme SSPI. So for this one, I actually couldn't find the second one. I typed all of the enzymes that was listed in this picture, but I only found one of them, which is at SSPI. However, for the organism I am wanting to insert my gene into, this is actually the wrong plasmid. But I figured I should just include this since I did do some of the research. But anyways, this brings me to my next slide, which is the correct plasmid. This is the plant plasmid, which is P5A1TI. As for the enzymes, I have PSII, and fortunately, I was able to use the same enzyme and cuts from the other plasmid slide. So that would be SSPI. Now as we can see here, I drew a circle around where the cuts are, and the color-coded gene codes correspond to the green and yellow highlights. So for enzyme 1, PSII, the code would be TTATAA in green, and for enzyme 2, the enzyme 2, SSPI. The code is AATATT in yellow. So to give a little bit of a background information on how this plasmid enzyme selection stuff works, at the origin of replications, the DNA opens and allows for replication to start. And then there is a multiple cloning site where the selected gene will be inserted into this plasmid. Once this happens, there are restriction enzymes that can cut the DNA and then it glues them back together again. But eventually the plasmid infects the organism of choice and will later show its physical expression. So while we're on this topic of organism of choice, my organism of choice, I wanted to use this bioluminescent protein as a way to help fix the dark streets. I couldn't think of a good organism of choice, so I just went with lemongrass. I thought the long leaf parts were pretty cool. But anyway, if we were able to do this project in real life, I would want to insert this gene as a test. If this gene that I insert lasts long enough in the lemongrass, it may be able to be inserted into other plants like grass or even flowers maybe. But the main end goal would be to light up the streets that have no lights. And also I hope not to kill any plants in the process or harm any plants in the process. Now for the transformation method, there is not really a picture I had for this slide, just this process with the arrows. But I'm just going to summarize the agrobacterium plant video to the best of my understanding. So basically the TI plasmid transfers into the plant cell where the wounded site is. And this TI plasmid, while in the wounded site, can induce plant cells to make food molecules for the bacterium. Here it would release the compounds in which the agrobacterium receives the chemical signal and activates another protein. Via the plasmid, the tDNA moves and begins to undergo bacterial conjugation. This tDNA then becomes integrated into the plant genome, which later allows this new gene to be expressed in the plant. The end result would basically be a genetically modified plant, or in this case, a genetically modified lemongrass plant. Now again, the main goal of this project is to light up the streets or areas that have no lights. And in recent years, we definitely have seen an increase in crime rates, but lately it doesn't seem to matter whether it is broad daylight anymore or if it's really dark or if there are people around anymore. But anyway, and how this experiment project would help is this may help with costs and energies, as these would not be using electricity. As for a con, since this is a bacterium, or it is a bacterium, but it's a marine bacterium, which is what leads me to why I said what I said earlier about testing its longevity. If this is a bacterium that normally survives in a sea organism, in seawater, will it survive on land? Will it need seawater? This would be one of my main concerns for this gene project if this were really to happen in real life. And lastly, these are my references. Thank you for your time.